Hello, welcome to the Web Groups webinar. I'm Mike Travers from the SRI Bioinformatics Research Group, and we're going to teach you about web groups, which is a relatively recent new feature on the BioPsych website. So groups are collections of BioPsych objects, such as genes or metabolites, that you can create. You can create them by uploading data from a file or from searches within Pathway Tools. Once you have created them, you can use transformations to explore relationships between your objects and other knowledge in the database. You can generate new groups based on those relationships, and you can perform a variety of computational analyses, such as enrichment analysis. So once you create a group, so groups are linked to your BioPsych account. As you can see here, this is the group directory. And we're not logged in, so what we're seeing here are public groups that other people have created and decided to make public. So for you to create a group, you have to log in, and you can log in the usual way. There's a special link here, and I will log in. If you don't have an account, there's a link that lets you create one right there. So once I log in, I will see not only public groups, but also the groups that I've created in the past. This is a tab of my groups. And so we will start off by creating a new group by uploading a file. So here's the file. It's a list of gene names. So we'll go to the Groups menu, say New Group from Uploaded File. Pops up a little dialog here that lets us specify the file, which is this one here. TRP, differentially regulated genes. And we will upload it. And there it is. So here's our group. Now a group is structured very much like a spreadsheet. It's got rows and columns. The intersection of row and column is called a cell. This group that we just created just has a single column, which is called column 1, but maybe we'll change that to something more descriptive by clicking on the title. You can change the names of columns and of groups by clicking on them. And it's got about 120 rows. So once we have that, we can create new columns, which are relationships or properties of the genes in the first column. So this menu here says Add Property Column. We can choose some properties of genes, such as synonyms. So that creates a new column, which shows other names for that gene that are in the knowledge base. Or we can pick the accession 1 property, which shows the accession number. Once you have these properties, you can do things like we can sort the genes by accession number, if that's interesting. Then the transformations work in a similar way. They create new columns, but they're based on a slightly higher level relationships with the knowledge base. So these are all the transformations we have on genes. We can, for instance, create a column that contains the product of genes. There's the enzymes there. And once we have a column like this or any other column, we can create further transforms based on that. Now notice that this first column is slightly darker color. That means it's selected. We can select another column by clicking in the header. So now we'll select the protein products that we created and see properties of that, such as, how about the molecular weight. So now we have a property that's not based on the gene, but on the protein of the gene, and we can create chains of these computations indefinitely. Let's see, one other thing you can do is once you have information in a column, you can paint that on the cellular overview. So let's do that. We can see where these genes we're interested in live. There we go. We can let's try show one other operation here. I'm going to get rid of some of these columns, which you can do by clicking on the little blue delete icon. Another transform shows the pathways that these genes appear in. There may be more than one. And once you have that list, you can create a new group with those objects by clicking on this green button. So that makes a new group, which is the union of all of the objects in that column, each one given their own row. And it also happens to show the matching object, the object that we started from, that caused this pathway to be in here, and sometimes there'll be more than one. Okay, let's go back to our group, and I'm going to show another thing you can do. Let's see. This is our group of genes, and we'll get rid of some of this. So enrichment analysis is a statistical technique that lets you, given a set of objects, such as genes in this case, will let you find related objects or concepts where those genes are statistically over or underrepresented. So the idea is that if you have a set of genes from an experiment, and you want to know if those genes share some Go terms in common or pathway in common, enrichment analysis can give you a rigorous statistical way of determining that. So we'll, in this case, we'll take our gene set and enrich that for pathways. So that pops up a dialog which lets us specify some parameters for how this computation is going to take place. We'll just take the defaults, click Go. We'll go think for a bit. So there we go. So now we have a list of pathways. So this makes a new group. Each row of the group contains a pathway, contains a p-value, which is represents the likelihood that that pathway appears by chance. So a low p-value means that's unlikely, so it means there's probably something meaningful about that pathway being associated with that gene list, and this group is sorted by p-values inversely. And it also shows you the matching gene that caused, or genes in this case, that caused these pathways to appear. And maybe I'll just delete that column for now so we can see more of the pathways. So these are the pathways that are overrepresented in that gene list, and since that tells us something about that list. Okay, so this has been based on experimental data. Let's try something else where we're just using groups to investigate the knowledge that's in the knowledge base. So let's say we want to find out something about the genes that are regulated by a particular protein. So first step there is to do a search for the protein, which we'll do there. And you'll notice here that there's buttons to turn the various search results into a group. We're not going to use those now. Instead, we're going to go to a page for the particular protein that we're interested in, ARCA, phosphorylated DNA binding transcriptional dual regulator, and we'll go there. And notice that so on this page and any other object page in the system, we'll now have an Add to Group button that appears on top there. And if you put the mouse over that expands a little bit, and it gives you some options. You can these let you take that object and add it to either an existing group that it knows about, or make a new group with just that object in it. So we will do that. We'll pick new group and say Add. So that creates a new group and gives us a link to that. We can click on there. Okay, there's our new group. It's got one object and one row in it. That by itself is not very useful, but we can do transformations on that. So let's see what we can do there. So here we can go to find the genes that are regulated by that protein. So like as before, this will add a new column. There it is. So that's quite a long list, and it's not that useful in that form because these genes are all clumped together in one cell, because the group still only has the one row that we started off from. But we can take that list and turn it into a group of its own by clicking on the green button. So now it's made a new group with all the 168 of those genes, each given their own row. It showed us the matching object, which is not that interesting. It's because that's going to be the same for each row, so we'll just delete that. This name is kind of unwieldy, so maybe we'll click on that and make it something more shorter, more descriptive. Genes reg. So we've pulled out this list from the knowledge base. So what can we do? What can we do with that? Well, let's do it. Let's do another enrichment analysis. Let's find out what pathways are overrepresented in that set. And as we can see, there's a certain set of pathways that are protected, respiration pathways, generation of precursor metabolites and energy. So that tells us a little something about the genes that are regulated by that protein. Another example where we build a group from the results of a search. So we'll go to the standard search page. We'll search for proteins of a particular. We'll search for short proteins whose amino acid sequence is less than 100 long. Do the query. There it is. And now we have a button on here to turn those results into a group. And there it is. So the group looks a lot like the original results, but now it's in a form where we can manipulate the data a little bit more conveniently. So, for instance, we could, let's say we're not interested in the product name. We could get rid of that. We could look at the position on the gene, sorted by that, if we wanted to. Or we could do enrichment analyses. We could find lists of genes in the same operon. Now we've got a list of genes that are in the same operon with a short protein. Let's do an enrichment analysis on Go terms for that. There we go. So this is interesting. So that's produced some definite results. Structural constituent of ribosome, structural molecular activity, RNA binding. So this gives us starts to give us some clues about what kind of genes are in our set. Clear that up and we can see them a little bit better. All right, well that concludes this webinar. There are more things you can do, and we will have a subsequent webinar that explains some of those things, which include manipulating DNA sequences within groups. Thank you.